Welcome to Vienna, European Hematological Association. I'm Gordon McVie and I'm the editor of ecancer. We've been filming a number of key opinion leaders in the corridors for the last day, and we're also doing some expert-to-expert interviews, and my expert today is Professor Simon Rule from Plymouth.
Good afternoon.
Thank you for joining us. Tell us about mantle cell lymphoma. Now, not everybody watching this program will know much about mantle cell lymphoma, and I suspect that the bottom line is that if they don't know very much, they need to refer a patient with mantle cell lymphoma to a centre straight off. So what happens when they come to you? What's given the EHA meeting abstracts, there are quite a few actually, there are over a dozen on mantle cell lymphoma. How are you going to manage things or recommend other people to manage mantle cell patients? Up front, first of all, new patients.
So first thing to say, mantle cell lymphoma is a rare disease, about 1 per 100,000 population. So in the UK, for example, we're talking 500 to 600 new patients a year. So it's rare, incurable, and of the lymphomas, it's an aggressive type. Yeah. We've got fairly good frontline therapies now, particularly combinations of chemotherapy and rituximab. Rituximab has made a big difference in older patients. And in younger patients, we tend to be a bit more aggressive. Different styles of chemotherapy consolidated with an autologous stem cell transplant. Rituximab maintenance now established in elderly patients, and data, very recent data showing that in the context of autologous stem cell transplant, rituximab maintenance adds progression-free survival benefit as well. So frontline therapy is fairly well established. When patients relapse, though, it then becomes a bit more difficult. Sure. And whatever you tend to use then from a conventional perspective, your responses are less adequate, so not as complete and usually shorter. The big revolution in mantle cell lymphoma over the last couple of years has been some novel drugs, and principal amongst those the BTK inhibitors, and they really have been transformational. And I suppose the interesting thing now is how we're going to incorporate these novel drugs into our frontline and subsequent therapies. So those trials are being run right now. There are other drugs. There are 4 drugs in the world that have a license in mantle cell lymphoma. So ibrutinib, which is the BTK inhibitor, is the most recent, but also temsirolimus in Europe, which is probably the least effective of the agents. Lenalidomide, and there's quite a lot of data on lenalidomide at this meeting, and Velcade, bortezomib. All those drugs have activity. I think what's interesting now is where we add these drugs into conventional chemotherapy. Do we use them instead of chemotherapy? A lot of questions, really.
So bortezomib is in frontline in 1 or 2 combinations.
It is. Velcade has activity in mantle cell lymphoma, clearly 30 to 40% as a single agent. If you add it to chemotherapy, you get an additional benefit. I've done that for a long time now. And there's a recent trial published this year showing that if you add Velcade into standard R-CHOP chemotherapy, well, it's not standard because you take the vincristine out, then you get a significant PFS advantage. So I think adding Velcade into frontline is close to becoming established. Mm-hmm. In every trial that's added Velcade to an established regimen in mantle, you've seen a benefit. So it adds something.
This is pre-transplant?
This is pre-transplant. This is predominantly in patients who are ineligible for transplants, the older patients.
And the costs, I thought I saw an abstract here on the costs of those frontline therapies. Because they're more expensive, but they give a longer.
Well, that's the balance, isn't it? You get more bang for your buck, and then, well, I mean, we can have a discussion about costs, and it applies to just about every disease, doesn't it? Because all new drugs are expensive, but you get a benefit for it, and I guess that's what government regulators are there to decide whether that's acceptable or affordable or not.
And patients who are not eligible or who can't have a stem cell transplant, is that the same kind of
that's the majority of patients. So, I mean, the average age of presentation is sort of late 60s, maybe even be a bit later looking at UK data. And for those patients, you know, if you're very fit, then having an autologous transplant is an option. But for the majority, you're looking at pretty much standard R-CHOP type chemotherapy, what one would use for aggressive lymphomas. Bendamustine increasingly being used, particularly in Europe and North America. And getting to be used more frequently in the UK as well. So, and those two are probably comparable. as far as efficacy.
So is that the setting where you're going to put the new drugs coming out of the maintenance arms? Tell us about the maintenance arm first of all, because there's a nice trial that you've been involved with.
Sprint was comparing lenalidomide as a sort of single agent against investigator choice single agent. So you're asking a question whether that, in a randomized setting, was better than what other single agent drugs we had available.
And resistant relapsing patients with mantle cell lymphoma.
Heavily pretreated patients, and investigator choice which of the single agents you were going to use versus lenalidomide. It was a 2-to-1 randomization, so more patients, 2 patients got lenalidomide to 1 patient getting investigator choice, and a clear benefit for the patients getting lenalidomide over any of the single agents.
And there were 5 on offer.
Yeah, and whichever one you used.
But the numbers in the 5 other must have been quite small because it was a 2-to-1 randomization.
Yeah, I mean, and that's the criticism. You know, you get into subgroup analyses, but overall there was a PFS benefit.
Well, the approach was, I think, quite novel, and I liked it a lot. And I think that the intention to treat shows that lenalidomide has definitely got an advantage. And you've got an abstract, I think, on quality of life in that study.
Yeah, so we looked at quality of life in that very study, and it showed that the quality of life wasn't, well, it's pretty much the same basically in both arms. And of course, the lenalidomide patients were treated for much longer, so quality of life was maintained despite that. There was some suggestion that there were some elements of quality of life that were slightly better in the lenalidomide arm, but it certainly didn't disadvantage patients. And that's a concern sometimes when using a continuous therapy, that might have an impact on quality of life in patients, but that's not the case with lenalidomide.
And now the question, what are you going to do with that then? Are you going to put that into the upfront with patients who are not fit for transplant, or are you going to go straight in for the younger, fitter patients? What do you think we should be doing?
So lenalidomide in combination, there are a number of combinations that have used that with other drugs. It's quite difficult to combine lenalidomide with other drugs because it's quite myelosuppressive. And I think that one of the troubles is these drugs are very good, but the BTK inhibitors are that much better. They've sort of trumped them, really. So they're twice as effective with fewer side effects. So the question is, what are we going to do with ibrutinib? And there are other BTK inhibitors. I was talking about those this morning at the Mantle Cell session. Ibrutinib, in the context of chronic lymphocytic leukemia, just shown a massive benefit in a publication last week at ASCT.
There.
No,
contest.
And those trials have happened in mantle cell lymphoma as well. The big question is, can we use that as part of frontline therapy? And we've got a trial. I've got a trial that's just about to open in the UK, which is going to compare ibrutinib with rituximab versus standard chemotherapy. So that's the only trial in the world that's going to do a head-to-head comparison of chemo-free versus chemotherapy frontline. So that'll answer the question whether it's better. And then the other issue is, how do you sequence these things? I mean, I don't think anybody believes we're curing people with these new drugs. And it is exactly how you sequence them. So you can certainly salvage anybody after lenalidomide with ibrutinib. That may not be the case the other way around. We don't know. We don't yet have enough experience with the new drugs when patients relapse, because they do seem to set off some resistance mechanisms that might make subsequent treatments sometimes tricky. So and it's difficult in a rare disease, that's the other thing, to do sufficient trials to actually answer the questions that you really want to know the answers to.
But strategically, there's some really strong national groups in this field, which is really, I think, a lesson for some of the other solid tumour disease groups. And also on the rarer tumours like mantle cell, there is really good collaboration across.
So this morning's meeting was the European Mantle Cell Lymphoma Consortium. So, you know, there's a good European collaboration there. And within that, actually, we've got a couple of trials running. So there's the major trial running between the major groups. And then I've got my frontline study, which we're doing with the Nordic Lymphoma group. Okay. And there are enough patients to do that. So yeah, I think you have to collaborate. You can't do these trials without big national groups getting involved.
So nobody's being cured at the end of the day. So what are the targets? I've seen some stuff on PRK, downstream targets and SOX and CD19. Where would you put your money?
Well,
what are you doing with all?
There are lots of targets. You know, the B-cell receptor pathway is clearly where the action is, and there are lots of potential targets within that. PI3 kinase perhaps isn't the answer for mantle cell lymphoma. These drugs are active in mantle cell, but the duration of response when you use these drugs is really very short. And I think that group of drugs discriminates itself for the more indolent forms of lymphoma. BTK clearly very active in mantle cell lymphoma. BCL2 inhibitors, possibly very, very active. Well, we know they're active. So it's going to be combinations is the answer.
Yeah.
I mean, I've got a trial that's going to open later this year that's going to combine ibrutinib with a BCL2 inhibitor with an antibody.
Right.
Now, whether if you combine novel agents that all have distinct mechanisms of action as far as killing is concerned, you then potentially deliver a cure, we'll see. I mean, if you follow a TB type model, then that's what you have to do. You have to combine these things. So that's the way things are going to go quite quickly. And what's interesting is that the drug companies have lined themselves up. They have interest. There are basically 3 BTK inhibitors right now that are being evaluated seriously, and all of those companies have PI3 kinase inhibitors and BCL2 inhibitors and PD-1. So you're going to see a number of combinations over the next couple of years, and we'll see. Of course, you never know when you combine novel agents whether you're going to get unknown toxicity. You know, of course, we've been bitten by that over the years in the lymphoma world. But hopefully combinations will improve efficacy without that.
And what about the whole CARs area, the chimeric antibodies? This is really, I mean, this is quite seriously, potentially difficult area, but also some very exciting preliminary results.
Yeah, I mean, fascinating. I was at a meeting in Chicago a couple of weeks ago, talked about CAR T cells, and yeah, I mean, they are. I mean, I'm going to have to go back to my medical school days and relearn immunology, because I think cancer now is all going to be about immunology. I mean, that's the way these things are going. Fascinating how these things work, but some quite frightening toxicity in patients. I mean, patients requiring elective ventilation is a bit of a concern. But when they come out the other end, I mean, you're curing some really, really difficult diseases. So I think if you can get the toxicity right, the trouble is people don't know what's actually causing the anti-tumor effect. So, you know, if you dampen down IL-6, for example, you might lose the anti-tumor effect. So, fascinating, absolutely fascinating. Nice to have a lot of potential drugs to play with.
It's a good time. Simon, thank you very much indeed. I really appreciate it. Thanks.